much for your kind introduction. Well, the good news, this room has black walls, so hopefully I'll do okay. And I want to begin by saying on behalf of all authors that we are extremely honored and humbled to have been selected for the award, since we know that the award committee must have had a lot of excellent papers to choose from. And it is my great pleasure to acknowledge my co-authors, Wes Solomon, Sam Jolie, Melissa Moore and Phil Zaymore, and Melissa at this meeting. So this was collaboration between Moore and Zaymore labs. And I just want to tell you, today I'm going to tell you about a couple of things that we discovered in our paper. These are rather fundamental things about Argonaute action. And one of them is really, how does Argonaute use the small RNA guide to find its target? This is not as straightforward a question as one might think, because as I'm going to discuss, the single-stranded RNAs have a couple of inherent problems that make them less than ideal probes for finding complementary sequences. And the other question we wanted to ask is about the seed region of the guide strand. As you all know, the seed region is a special region in the small RNAs that is important for microRNA binding. And so we wanted to answer the question about the function of the seed, how it is formed, and what it does really. So let's start with the first question. How Argonaute uses the guide strand to find its target? Well, indeed, the short single-stranded RNAs have a couple of inherent problems. And first one, they make extremely stable complexes with complementary strands. So this high binding stability actually translates to low binding specificity, because the dwell time or the lifetime of this duplex before it spontaneously dissociates can be staggering, extremely long. In fact, it could be 10 to the 10th years for duplex this length. And introducing each mismatch that you introduce to this duplex will shave off 1 or 2 zeros of this figure, but because there are so many zeros, even many mismatches will still bind tightly. And the other problem is that homology search by RNA oligomers is really slow. We know that their theoretical limit for this process, this is essentially the diffusion limited rate of bimolecular collisions, and this rate is about 10 to the 8th to 10 to the 9th per mole per second. But the actual RNA hybridization rate is about 1,000 times slower. So only one bimolecular collision between complementary strands will result in hybridization. Well, this has to do with entropy, and because single-stranded RNA is so flexible and samples a lot of conformation, most of these conformations are not conducive to duplex formation. So this process is extremely inefficient. And so today I'm hoping to show you how Argonaute solves all these problems. And none of these questions can actually be answered by the known hybridization rules. And to answer these questions, our favorite approach is to use single molecule imaging, and we do believe that this is the best way to do quantitative biochemistry. So here we have our fluorescent target molecule, and we have fluorescent RISC, but target and RISC are labeled with different color fluorescent dyes so that you can tell them apart. And each of these molecules look as a diffraction-limited spot of light on your images. And if you have two colors colocalizing at the same time, you know that you have binding of RISC to the target. You can also observe RNAi in this format. You can actually detect splicing. So this is one of the typical movies we collect in our experiments, and the RNA targets are in blue color, and RISC molecules are in red. So whenever you see magenta, that means that RISC is bound to the target. And I hope you can see that at first there's a lot of target molecules, and then they gradually disappear. And each disappearance actually is preceded by the RISC binding event. Well, perhaps it's easier to just look at a single target molecule. Here we have a single target. Then RISC comes, binds, cleaves the target, and it disappears. So effectively, we can observe all these reaction species in real time, and we can get all the kinetic and thermodynamic parameters we possibly need from this simple experiment. And one of the most useful parameters you can get is just the binding rate. You can get it by looking at hundreds or thousands of molecules. So this curve, this binding curve, is for LED7 guide strand. There's no AGO yet. Binding to its complementary target, and the rate that we detect is consistent to what I said before. This is about 10 to the 6th. But what about LED7 AGO? It turns out that LED7, that Argonaute accelerates binding of the guide strand by about 2 orders of magnitude. And at this point, we realized that actually LED7 is a 3-letter sequence. It doesn't have a C, and so we wanted to look at other 4-letter sequences like MER21. And we know that 3-letter sequences actually hybridize faster because they cannot form a secondary structure. And indeed, 4-letter sequence binds slower, but what about the MER21 RISC? Again, we see the same acceleration. In fact, it's even greater. It's faster. It's 250 times faster than the free guide strand. So next, we wanted to ask whether this acceleration is actually conserved among Argonautes. So on the left is the mammalian AGO we have been looking so far, and on the right is the prokaryotic AGO, and these two Argonautes are separated by 2 billion years of evolution. And from some unpublished work from Seymour Lab, we know that TT-AGO actually uses DNA guide strand, and also the guide strand is a bit shorter. It's only 16 nucleotides. So we wanted to do the same experiment for TT-AGO, and at first we measured just binding of the DNA guide strand. We did it for both RNA and DNA targets simply because we don't know what the native target is. The biological role is not known. And then we measured binding of the TT-AGO RISC to these targets, and we see exact same thing. So this acceleration feature is highly conserved among Argonauts. So next we wanted to sort of dissect the guide strand and understand where this acceleration resides. So we made targets that complementary to either 5' half or 3' half of the guide strand. So we found that actually this acceleration comes from the 5' half of the guide strand. And dissecting it further, we found that actually it resides in the seed region. So how do we explain this data? We were lucky because about at the same time, high resolution structures of mammalian Argonauts came out of Ian McRae's lab. So here's human AGO with bound guide strand. So this is the seed region. And except for the first position of the guide, this entire region is in perfect A-form RNA conformation. So what Argonaute does is essentially prepays the entropic penalty. It pre-organizes the seed, and it keeps it in a perfect A-form conformation, ready to base pair. This is where the acceleration comes from. So we wanted to dissect it a little bit further and created like a set of mismatch targets with mismatches spanning the seed region. So we found that the target complementary to the seed region binds just almost as fast as perfectly complementary target, but mismatches in the region of the guide strand in positions 2 through 5 result in large defects of binding rate. And from now on, I'm going to call this region the seed core, because it's going to be important later on. And the rest of the seed seem to have almost no effect. So we have these two subdomains here. Next, we wanted to use the same mismatches to now look at the on the target departure and the dwell time, how long RISC remains bound after it finds the target. So again, the target complementary to the seed actually is bound very tightly. It stays for almost 5 minutes, which is, by the way, 25 times more stable than a guide alone would stay. And all the mismatches in the seed core result in very dramatic defect in the dwell time of RISC. In fact, by an order of 3 magnitudes. But as you move the mismatch towards the 3' end, you see smaller and smaller effects. But they do tell you that you need complete complementarity to the seed to actually ensure fully stable binding. So we can also look at the free energies, and this is interesting because we can predict free energies based on hybridization rules. So this is the prediction for LAT7 sequence. It would be different for different sequence. But what do we get when we look at the seed core region? We get absolutely flat profile. It does not follow the hybridization rules. And in fact, it looks like a single recognition unit. So none of the positions are more important than others. And it looks like a single RNA recognition domain. On the other hand, the 3' portion of the seed does follow the hybridization rules. And in fact, if you recall the crystal structure a few slides back, that part was not even visible in the structure. That's because it doesn't make contacts with Argonaute. Therefore, it behaves like free RNA, and therefore, it must follow the hybridization rules. And you can see that it does. So we can further explain some of the features that are shown by the crystal structure. So if the crystal structure, you can see that actually you can base pair the seed core to the target up to position 5 without inducing any conformational changes in your protein. However, if you want to base pair beyond that, this alpha helix needs to to swing away, and this induces a major conformational change in the protein. So in other words, this region up to position 5 serves as a fast homology finding domain, and the rest of the seed is required for everything to click into place and to ensure stable binding. So to summarize, we found that RISC binds target very fast due to pre-organized seed, and the seed has subdomains with very distinct function. We actually found that the positions 2 through 5, which we now call the seed core, are important for fast homology search, and the rest is needed for stability. Argonaute essentially make the guide behave like a protein RNA binding domain, and none of these can be predicted by hybridization rules. Finally, I want to really quickly point out that that this homology search mechanism is very common for essentially all homology search going on in biology, like homologous recombination or CRISPR-Cas9. In this example, you see Cas9, and for Cas9, it also looks for microhomology first. So there's a domain looking for PAMs, and that's your analog of the seed core. So once the PAM is found, all the subsequent stages or checkpoints, if you will, become slower and slower, and the commitment for stable binding is increased with every checkpoint. And finally, I did not have time to tell you about maybe equally interesting and important findings of the paper that have to do with RNAi, but I do hope you read the paper if you haven't done so. So with that, I just want to again acknowledge my co-authors and all the people who helped us along the way, and thank you very much for your attention.
Great paper, congratulations. The question is, now we are learning so much about the 5' phosphate.
Yeah.
If you do the same thing with the 5' phosphate, do you see any differences with and without the phosphate?
Yeah, so we never tried to actually omit the phosphate because what we do know, it's not supposed to work without the phosphate, but we did find interesting things about, for example, the position T1 of the target. It seems to be actually important. So the adenine in position T1 of the target is recognized by AGO. But we haven't explicitly looked at the role of the phosphate. It's actually a very good idea. Yeah.
Yes, great paper. Wes mentioned to me that this phenomenon of accelerated hybridization by pre-organization, that you somehow showed that this really accounted for the advantage of RNAi, that there was enough to explain the enhanced potency within the cell of an RNAi versus a free antisense. Could you talk about that?
Yeah, absolutely. So this tells you that all the therapeutic oligos they are don't work as well as they could work because RNA or single-stranded nucleic acid, for that matter, has these inherent problems. So Argonaute effectively is great at guiding RNA oligos to their complementary sequences. And we're actually looking into some applications of this, especially because, as you might know, the guide strand does not have to be 21 nucleotides. It can be longer and have some interesting, useful features. Yeah.
So one of the themes for the meeting has been the chemical modification of siRNAs and how that plays into RISC. And further, I'm stretching it even further, how chemical modifications can be placed into mirror mimetics, and whether the structural information that you've now obtained will give us some insight into A, why certain modifications are acceptable, and B, how modified can a mirror mimetic be in order to make those more effective drugs?
Yeah, that's exactly right. And now we are trying to turn this technique into something compatible with our research pipeline, where you can very quickly look at all kinds of modifications. In fact, we started some work with Anastasia, but we just started this work. Yeah.
Thank you.
Okay, please join me in congratulating Victor.
Thanks. Thank you.